And now I want to present what we have done. It's a paper that we have done quite some time ago, and where we compared the mycorrhizal communities in different land use systems. So it's fairly far away from the one-to-one interaction, but it fits quite well into the last work package of the project. We are working with mycorrhizae, which is the interface, or which sit at the interface between plants and soil, and they help the plant acquiring mineral nutrients like phosphorus and nitrogen, and they consume plants' carbon. And in this study, we actually used all three. We quantified all three pathways using radio and stable isotopes. So phosphorus has only radioisotopes that could be used for tracing, and for carbon 15N, not 15P. And nitrogen and carbon have the stable isotopes that could be used. So it's 15N. It was a rather complicated design where we compared soils or communities of microbes in soils from cropland, so from agriculture used lands, and from grasslands, so from lands or from soils that have sometimes been used as a pasture or as for hay production, but basically was not plowed for several years. And what you see here is that basically the brown color stands for the cropland, the green color for the grassland soil, and then we've collected soil. We collected pairs of soils from 4 different villages, and then that was actually complicated because then you get the soil and then you have the microbes inside, and you need to separate them. They're not easy to culture, so you have to work with like complex communities. And now the question is how to do such an experiment. So the experiment, and of course every soil is different. So every soil has different properties, different sorption, desorption properties. It has different water holding capacity. So not only the microbes. So we tried to make to arrange the experiment so that we could have the soils with the microbes, except the mycorrhizal fungi, because we know that's a microbial guild that is of particular interest, and we wanted to quantify it. But then we had to make a lot of treatments actually when we returned the original microbes from the cropped soil of this village to the same soil that has been previously sterilized and enriched with microbes without mycorrhizal fungi, or we used the same soil that has been sterilized, but we put the other inoculum in it, the other mycorrhizal communities. And now the point is how to sterilize the soil. That's a difficult task, actually, to sterilize soil. You can put it to autoclave, and then you mess up everything. If you autoclave soil, you will basically cook it up. So we'll denature everything, denature proteins, you will boil, you will degrade many structures. So that's not an option that actually is good for anybody. It's probably fairly good to get rid of some microbes, most of the microbes, not all. But we took another option, which was gamma irradiation. So we gamma irradiated soil, which is also has drawbacks. There are actually bugs that can survive quite high doses of radiation. I mean, cyanobacteria can survive incredible doses of radiation, but we didn't really care too much about that. So we grew plants either in soils that have been deprived of the indigenous mycorrhizal fungi, or that have been where the mycorrhizal fungi have been put back, or where basically they have been cross-inoculated with the other fungi which were adapted to the soil. Actually, this picture depicts it. So you see the grades of the colors. They somehow signal these are different soils. You cannot really cross this very much. You cannot take this fungus and bring it to another place, because then it is an invasive research. That is invasive biology, because that's really bringing it to a different realm. But actually, when you go to the same village, and then you just go, you know, at the border of cropland to grassland, you cross a few meters, then you're actually there, and then you have the microbes from one system. You can cross-inoculate to the other, and vice versa. So we had a lot of treatments, So we basically wanted to test the effect of the land use of the microbe of the mycorrhiza origin, but we had to also take the painful way of working with different soils and actually create the relevant controls. And we grew plantain, Plantago lanceolata, which is a plant that is pretty common in various systems. It's also pretty good model plant because it's not too big, it's mycorrhizal, and we created systems where we could grow the plant in the different soils with the different fungi. So the plants were colonizing the soil volume quite efficiently. And then in the middle of the pot, we had a compartment that could not be colonized by the roots, but only by the fungi. There was like semi-permeable membrane that has pores of the size that cannot be grown through by roots, but only by the hyphae. And this was equipped with a shaft with basically a pipette tip, where we injected the soil nutrients, to the nitrogen and phosphorus, or better said, the labeled nutrients. If you look who is actually growing in these systems, that was actually interesting because we could get quite a consistent picture across the different villages. You see these pictures are relatively close to each other apart from this, where you have most of the bars, or the gray bars are bigger, the green bars are bigger, and the brown bars are smaller in the grassland compared to the cropland. And this is also mirrored in this multivariate analysis. You see that basically the green spots, they somehow get together in the right hand side, and then the brown spots, that means the different croplands, they concentrate in the left hand side. So it seems that really this is the factor that makes the difference. So we're quite happy to see that there were, in spite of this coming from different villages, from different soil types, actually the land use over the past few years or decades has made quite a consistent difference. But this was not the ultimate goal of the work. Actually, that was whether the function was different. And it took us a long time to decide how we want to present the data. So finally, we presented the data as relative size effects, where we compared the size of the different mycorrhizal treatments to the non-mycorrhizal control. So this picture actually shows various information. So that's actually the position of the effect on the scale from minus something to plus something, with a 0 indicating that the plants were not affected by the mycorrhizal presence or absence. If the values were smaller, that means the mycorrhizal plants were smaller, or the values were smaller. And then the other thing, they always, you see, there are pairs of the values with some variation for each particular variable. And we could test whether these variables actually differ as depend on the land use. So whether the field was cropped, or whether the land was left as a grassland. So this is how the plants grew. You see many parameters, particularly for the phosphorus uptake, and this is known. I mean, mycorrhiza helps the plant gathering phosphorus, but also in terms of the plant size. Actually, we could see there were positive effects, so they are on the positive side. And what was probably even more important, consistently across all these different treatments, the plants that were that had the counterparts from the cropland, from the grasslands. So the grasslands were the green, croplands were the brown. They were bigger, both in terms of the shoots or roots, and there were also some other effects going in the other direction, whereas some other effects were not significant across the different land use histories. So you see, you can read many things. You can read the absolute size effect, but you can also see the differences. So here we got some indication that actually both mycorrhiza is good, But in some aspects, it's the grassland which is making a bigger effect on the plant, and some other aspects like the phosphorus concentration in roots and shoots, it's actually the cropland mycorrhiza that supports higher values. When we look at the isotopes coming up from soil, so phosphorus moving from the soil via mycorrhizal networks to the roots and to the shoots, you see very high values. But there were some differences, actually, but they didn't come up in these relative comparison, but only when we compared the absolute values. And there were also some indications that there was less nitrogen in the shoots in the mycorrhizal plants than in the non-mycorrhizal plants, which has something to do, I will talk about this later, I will show it later, that the nitrogen is immobilized in the fungi. And the carbon, that was just shaky a little bit. What was the most important thing is actually there was more carbon, so the values could be positive or negative, and this is not really easy to interpret. But you could see there was quite a big difference between the carbon that landed in soil, and it was actually more for the cropland microbes than from the grassland microbes. So they could they could drain more carbon from the plant to the soil, and this was actually significantly higher for the cropland. And getting to the end, this is how the nitrogen actually looked like. For the nitrogen, we could do a full account because we know how much of the N15 we put in. So we put about 100% of the N15. So this is calculated per percent of the nitrogen added. I mean, we have some ideas flipping over the chart, but this is probably just the experimental variation. But what's important is that for the non-mycorrhizal plants, non-inoculated or non-mycorrhizal ones, there's actually a lot more N hanging in the soil at the source, so not being moved anywhere. And actually, there was less hanging in the roots and more in the shoots. And this is showing that the nitrogen moves a different pathway than from the mycorrhiza, because then the nitrogen moves away from the spot where we put it, but actually there's a lot more hanging around in soil. That's also significantly more in the roots and actually very little in the shoots. And this is what mycorrhiza does. It does slow down the transport from soil to plant. It will eventually land in the plant, but it needs to be released. And actually this is what mycorrhiza does. It doesn't like open the valve, it just opens a little bit and then waits what it gets in return. So this is a nice actually show or nice illustration what mycorrhiza could cause. Not always the effects are entirely positive when we look at one aspect, but then you look, there's something happening with the nitrogen being redistributed. There's a lot less left when we put it, which is quite important. What is also quite important is that this total amount is lower in these treatments than in the others, which means that some nitrogen has been lost. And this is a big problem with nitrogen. Nitrogen is being lost. A huge amount of nitrogen fertilizers, which are costly and that are brought to the fields, they're simply lost either as gas or as a solution. So this is quite an important one. We could do a similar thing for the phosphorus, but we could not do the same thing for the carbon, because then we would have first to measure how much has been fixed, and that's not a trivial task. Right, so this is what I wanted to present as something that we do at the very high complexity level. Also showing up what tools we're using. So we like to use tools like radio and stable isotope for quantifying fluxes in ecosystems or in plant microbial systems. And we are particularly interested in the mycorrhizal symbiosis. So if there are questions, I'll be happy to discuss. And sorry again for the troubles with the Zoom. I apologize for that.